Okay, I know I'm standing between you and lunch, so I promise to be prompt. My name is Morgan Fitzgerald. I just finished my first year in Dr. Bradley Wojtek's lab at UC San Diego. I'm wondering if I get a prize for perhaps coming the farthest to get here. But today I'm going to be talking about cardiac signal interference and building off of Fabian's work in terms of simulated signals. And so I asked to go after Fabian because I knew he would do all the heavy lifting in introducing some of these topics to you today. But just a recap of what we talked about in terms of this, there's this aperiodic phenomenon when you're looking at EEG neural data. They call this the 1 over F, this broadband background activity. And then the periodic oscillations manifest as bumps over this 1 over F background. And originally came out of one of the papers in the Wojtek lab and has been highly replicated in the last couple of years, is that there seems to be this flatter spectrum, the slope in older adults and then steeper in younger. And so what's interesting is a year ago when I found Fabian's preprint was that I was curious to the fact that it seems like maybe these traditional methods are not actually able to fully remove this ECG artifact, this cardiac artifact from neural data. So maybe we're over-interpreting EEG data as neural and we're not fully considering what's noise, noise contributions at least. And so as Fabian was showing, this is EEG signal only, and you're seeing when the ECG is not rejected, it looks very similar to the ECG rejected, and this is the ICA analysis. And there's old and young, old in the pink and young in the orange. And so when you look at the ECG components, we also know that these also have this aperiodic spectrum, as Fabian mentioned. What that means was a good question, but we know that they're demonstrating these age-related changes in aperiodic slope as well. And so that brings us to this question that was already introduced as well, of it's really unclear of whether these two signals are influencing each other or perhaps they're representing this parallel physiological changes associated with aging. And so what I was hoping to do with this project is to assess this, cardiac interference in EEG. And the way that I am trying to go about this, and it's very preliminary and still in progress, but is to first characterize ECG waveform shape really robustly, and then actually generate a digital twin. So be able to take real human ECG data and then digital twin old and young to generate a time series, and then be able to ask questions about how this time series is influencing simulated aperiodic data. And so in order to do this, I actually spent the last year with my coworker building this Heartbeats toolbox, which is a Python toolbox for ECG waveform shape parameterization. And so my lab had previously built a toolbox called Bicycle for neural oscillations. And what this looks like, this is just an example of an EEG theta oscillation as demonstration. And we find the zero crossing thresholds, which are the blue dots, as well as the peaks and troughs. And then this allows us to essentially punch out each individual peak waveform and then really characterize, over-parameterize, if you will, the waveform shape. And we get out this really nice data frame for each bump in the time series. And so what I did was I took this and I adapted it to work in ECG. And so this is just an example of parameterizing the R peak, but we're able to do this for each heartbeat for each individual. And we do this for all 5 peaks. And so we get both Gaussian and shape parameters. And then we get this really nice data frame with several components per all 5 peaks per cycle per heartbeat. And so what's nice about this is that the ECG is physiologically grounded. We know what's happening at each peak, rise, fall, trough in the tissue itself. And so we also know that, canonically, this amplitude and duration of the ECG signal, known with associated changes in the heart, are actually changing with age. So some common ones you're probably familiar with are R wave amplitude, as well as this QR duration increasing. And so one paper found that these features, just amplitude and duration, are actually able to predict ECG age. So think age in terms of you have a wider R, would be an older ECG, is correlated with this actual real chronological age. And so we took our tool and we were able to replicate age expected effects. So you're seeing QRS duration go up, you're seeing heart rate variability go down. This is an example of just the P peak, but you're seeing P peak duration go up, as well as this is reflected in our rise decay times. So this is a sanity check. And so now we have this, we think we're capturing waveform shape of the ECG, and what are we doing with this? Well, now I'm simulating a high R versus a low R based upon real human ECG parameters, and then I'm looking at the power spectrum of just the ECG. So how is this changing? And then I'm replicating this across all these shape parameters. I'm still working on stepping through this, but these are some examples, and you can see that we're changing the offset as well as the taper at higher frequencies. And so once we have the ECG time series simulated, what I then did was add this just with a linear combination into EEG aperiodic simulations with a package called NeuroDSP. This is just brown noise. And so with this linear combination, we're just getting a proxy for maybe how these signals are interacting in the power spectrum. And so this is the most drastic example of the change in the power spectrum, where you see the light blue is this ECG aperiodic background. So this would be like considered a purely neural signal, a purely neural background signal. And then you're seeing it added with the low R versus the high R and how it's changing the spectrum. And so I've also replicated this across the same other waveform shape parameters that I just showed you. We're not seeing super drastic changes with these ones, but as I said, I'm still working on stepping through all these waveform shape parameters. And again, this is just a proxy for how these signals might be interacting. So future directions, we still have this question of how these signals are maybe changing with age, how the tissues are changing in parallel, or maybe interacting. And so the next step that I'm working on right now is actually analyzing the simultaneous recordings in real humans of ECG and EEG, and then replicating this like digital twin idea in real human data and asking about age effects. And thank you.
Thanks a lot, Morgan. Any questions? Yes.
Yeah, thank you. So I think this talk is very good to address an elephant in the room, which we discussed with Marcel also yesterday. So when we talk about ECG, it's not just one signal, right? So heart is a rotating and moving dipole. And what is even worse, your chest is changing, so meaning your lead field matrix is also changing. So basically, have you considered taking more than one ECG lead? Because nowadays we use 16 ECG leads, also covering, let's say, back, neck, and so on. And actually ECG looks quite different, and spectral properties are quite different. And how are you going to account for this? And whether it's in your pipeline, so to say.
It's a great question. So yeah, this is based on 12 lead as well. We've looked at 2 lead and 12 lead so far. We're still working on getting the official like associations with age in those. We also have actually a partnership with the UC San Diego like director of pacemaker services, and we're getting pacemaker data like directly from the heart itself. And it is absolutely on the docket for us to look at all these different waveform shapes and the different heart signals. Absolutely.
Very good.
Maybe next year.
Nice, nice.